these are some summary slides to remind us of what we've already discussed or introduce some of these concepts again. Immunity is both innate and acquired or adaptive. The innate or non-specific factors are those types of factors such as physical barriers or chemical structures that simply provide our first-line protection from unwanted infection or growth of possibly abnormal cells. The acquired immunity can be either active or passive. Acquired immunity, again, this is where we're eliciting the involvement of the immune system. For active immunity, our own body will be making immune factors, or our own body will be essentially activating our own T and B lymphocytes for a specific immune response. For passive immunity that's acquired, that means some other source, either animal or human, will provide the immune factors. Our own body, our own T and B lymphocytes will not make the immune factors. We have to get it somewhere else for passive acquired immunity. Active acquired immunity can become natural or artificial. Natural active immunity, remember, active immunity, our own body, our own T and B lymphocytes are providing the response. So, for natural active immunity, that's where we accidentally get exposed to an infectious agent. Oops, we got sick. And truthfully, this is probably the best way, if you can survive, to generate an immune response and immune memory. The best immune memory is that which our own body produces our own T cells, and we have our own pool of memory T cells, and we activate our own B cells, and we have our own pool of memory B cells to confer immunity. So the best, if we can again survive the onslaught, the best form of active natural immunity is our best defense. But sometimes the pathogens are too powerful, and our odds of surviving are very low, or maybe none at all. That's when we take advantage of artificial active immunity. Remember, active immunity, our own T and B lymphocytes are being recruited and activated. But for artificial active immunity, we're deliberately exposing ourselves to some portion of a toxin that's been modified into a toxoid, or a bitter piece of a virus or a bacterium. That's why we have vaccines or immunizations, to elicit a strong, powerful, active immune response that's artificially triggered. For passive immunity, where some other source is giving us or providing us with some type of immune factor, which is usually antibody. We have both natural and artificial as well. For natural passive immunity, those are often examples of maternal antibodies transferred to the baby during pregnancy or after delivery in mother's milk. Antibodies provided in body fluids, anything that the mother is immune to by antibody response, then the baby is also going to have that protection passively through the mother's own immune factors. For passive artificial, that's where we have injections of antibodies or other types of materials that are immune factors. But again, they're usually antibodies that are injected. And antibodies from various types of sources. Human is best, but we even have antibodies that are produced in horses or other kinds of animals that can also be used and injected into a human being to provide some, again, passive artificial immunity. A passive immunity, unlike active immunity, since our bodies do not make the immune factors for passive immunity, these are temporary. They're not going to provide lifelong or long-term protection. Sometimes we just need a temporary protection, maybe for a few weeks or months. And that's where passive immunity can provide us at least some protection and help to get over or become well again. T and B lymphocytes have what we call receptors or markers on their cytoplasmic membrane that are capable of interacting or what we call recognizing antigen. On B lymphocytes, these structures are antibodies. And in fact, the antibodies are usually produced by B lymphocytes, and these are exported products. These are secreted proteins that wind up in our body fluids and tissues. T cells, on the other hand, have a membrane-bound structure that's called a T cell receptor. Both TCRs, T cell receptors, and antibodies are capable of essentially attaching to or directly interacting with antigens and are responsible for activating or eliciting that powerful humoral and antibody-mediated or cellular-mediated immune response that hopefully will eliminate and cure us of disease or infection. In summary, stem cells in our bone marrow, hematopoietic stem cells, through cytokine and interleukin activation, transform into T or B lymphocytes. B lymphocytes emerge from the bone marrow fully functional, but T lymphocytes must be educated or further activated in the thymus. That's why we call them T lymphocytes, thymus-derived lymphocytes that in fact respond to various types of tumors, pathogens, or other unwanted materials. T cells have TCRs on their cytoplasmic membranes that interact directly with antigen or foreign bits and pieces or non-self materials that are packaged in MHC markers. Once activated, these immature, now activated cells of T lymphocyte derivation produce either pools of memory cells and CD8 positive cytotoxic cells, CD4 positive helper cells, and there's a small pool of lymphocytes that also become natural killers. Natural killers are often more non-specific in their function than the CD8 positive T cytotoxic cells, but both natural killers and CD8 positive cytotoxic lymphocytes have the same killing mechanism of destroying our own eukaryotic cells that are infected with a virus or bacteria. Activation of CD4 positive cells, depending upon the type of communication, will lead CD4 positive Th1s to help macrophages or other APCs attack and eliminate intracellular pathogens. CD4 positive Th2s, on the other hand, are going to enhance B cell function, and in fact, CD8 positives can also assist once the target has been attacked by antibodies. We have essentially antibody-mediated or antibody-assisted or antibody-dependent cell cytotoxicity through CD8s working with antibodies to eliminate virally infected cells. Again, pretty complex system, and this is the simple version. So what do CD8 positive T lymphocytes do? They target intracellular viruses, intracellular bacteria, and even tumor cells, abnormal cells of our own bodies. What do CD4 positive cells do? They target intracellular bacteria, like the one that causes tuberculosis. So the Th1 CD4 positives targeting intracellular pathogenic bacteria. And what do Th2 CD4 positive cells do? The Th2 CD4s enhance antibody production by augmenting or eliciting a more powerful response in B lymphocytes. Antibodies are produced by B lymphocytes. And we're going to find there's also another type of cell that produces antibodies that are derived or produced from activated B lymphocytes. It's called plasma cells. There are 5 major classes of antibodies produced by B lymphocytes and plasma cells. They're IgG, IgA, IgM, IgE, and IgD. The Ig, capital I, lowercase g, is an abbreviation of the word immunoglobulin, referring to proteins, globulins, that have an immune function. Immunoglobulin is just another word for antibodies. Notice that in the illustration, not all classes of antibodies have the same appearance. There are some antibodies that are what we call monomers. They simply come in that simple Y-shaped structure. This is actually a simplistic version or view of the structured antibody. It's a little bit more complicated than this, but for discussion and illustration purposes, this common Y-shaped illustration for the monomeric form of antibody of any class is a very typical illustration that's uniformly viewed in every textbook. IgG, non-secretory IgA, IgD, and IgE are all found produced in the body in monomeric form. IgA is what we call a dimer. It's formed from 2 monomers of IgA combined or attached to one another with a J chain, which also includes a secretory piece, because secretory IgA, as the name implies, is the class of antibody that's found in secretions, tears, saliva, and any fluid coming from a mucous membrane. IgM, on the other hand, is called a pentamer. Pentamer meaning 5. We have 5 monomers of IgM that again are linked together, including the use of a J chain, to make this sort of starfish formation of the pentameric form of IgM. Think of the word gamed, G-A-M-E-D, to remember the 5 major classes of immunoglobulin or antibodies. The antibody class IgG is in the highest concentration in our body fluids like plasma or serum. Plasma is the liquid component of blood that has all proteins intact. If we remove the clotting factors from plasma, then what remains in the liquid form is called serum. And so oftentimes you hear the phrase serum antibodies, because serum is much easier to work with than plasma. The problem with using straight plasma to do any kind of analysis is that if you activate the clotting factors accidentally, then you have this sort of gooey, gel-like mess or mixture, and you don't want the gel getting in the way. So we often remove the clotting factors from plasma, producing serum, and then we analyze the serum antibodies from patients to determine their health or illness based on concentrational amounts of antibodies like IgG. IgG comes in subclasses. We have subclasses of IgG 1, 2, 3, and 4. IgG 1 and 3 are in highest concentration in placenta and can passively transfer from the placenta or in the placenta from the mother's maternal circulation into the baby. So IgG 1 and 3 are in fact the classes or subclasses of IgG that confer passive immunity in a developing fetus. IgM, the pentamer, is a glutenator or clumper. This is often the antibody class that's first produced in our earliest response to a pathogenic microorganism. First response, called primary response, IgM, first antibody on the scene to try and grab up and eliminate any of that unwanted bacteria or even virus. IgE is the major class of antibodies that works with eosinophils and basophils for allergic reactions that we call type 1 hypersensitivities. Type 1 hypersensitivities are called immediate hypersensitivities because when you're first exposed to the bee sting, the drug, allergen, within a few minutes you start to respond. In fact, the most severe types of type 1 hypersensitivities are called anaphylactic reactions, or anaphylaxis. The immune factors are activated so quickly because essentially the eosinophils and basophils are quick to respond and already have IgE attached to the eosinophil and basophil cytoplasmic membrane. So all it takes is really the antigen, a bit of pollen, a piece of the drug, the bee sting venom, to attach to the IgE already attached to the eosinophil or basophil to activate those granulocytes. Think of it as a loaded gun with the trigger already pulled. Once that trigger gets released, then you have the action or the reaction of that bullet speeding out of the gun and then of course causing that unwanted damage. Same thing is similar to an already sort of pre-activated eosinophil or basophil. The IgE is right there attached to the B basophil or eosinophil, and when you attach the antigen to it, it triggers that immediate response. Anaphylaxis can be very serious and of course life-threatening. Patients that are susceptible to anaphylactic reactions typically are required to carry an EpiPen or a source of epinephrine that can immediately counteract the immediate release of the vasoactive amines from eosinophils and basophils to prevent any lethal effect of anaphylaxis. IgA is the type of antibody, especially the dimeric form or the dimer of IgA, found in secretions from mucous membranes, tears, saliva. Any kind of secretion from a mucous membrane will have some IgA. So IgA is one of our specific immune factors that work with our first-line defenses, particularly in those exposed areas like mouth, nose, eyes. Very important when conferring immunity or protection from unwanted infection. IgD is our last remaining antibody class that really was mysterious and undiscovered for a long time. Antibodies are easy to work with since these are serum proteins that can be easily extracted. In fact, were some of the first immune factors from the immune system to be discovered and studied. We know a lot about antibodies. But IgD remained sort of hidden until further study of different types of diseases revealed the presence of these, what are really membrane-bound classes of antibody. One of two forms of membrane-bound antibodies, IgM, is one type. So M-IgM is membrane-bound IgM, but we also find membrane-bound or M-IgD. Membrane-bound IgD serves as a receptor and is part of the early activation of B cells. along with activating IgM. Sometimes it's left out of our discussions of activating B lymphocytes in an immune response, but IgD plays an important role in the very early stages of B cell activation. B lymphocytes produce antibodies, but plasma cells do too. Where do plasma cells come from? Well, as it turns out, plasma cells are produced from more differentiated and interleukin-induced activated B lymphocytes. B lymphocytes, upon first encountering any kind of bacteria, toxin, or in general what immunologists call antigen, those antigens can attach to the antibodies found on the B lymphocyte membrane surface, the IgD and the membrane-bound IgM. In addition, B lymphocytes serving as APCs will process and present bits of antigen in class II markers on their own cell surface by either directly interacting with antigen through membrane-bound antibodies and also interacting with CD4 positive Th2s. Interleukins, which are chemicals generally called cytokines, so cytokine is a general term, interleukin is a more specific type or example of a cytokine. The interleukins act as chemical signals to trigger the B lymphocytes to undergo differentiation. It's not all B lymphocytes, just the activated ones. And in fact, even the activated ones don't all turn into plasma cells. But we'll see and talk more about this in just a few minutes. If the animation runs that's on this slide, simply let it run through the whole process where you can see B lymphocytes activated and transforming into what we now call plasma cells or plasmacytes. Plasma cells are antibody-producing factories. Some of the change that we see in the cytoplasm of B cells that are activated and transformed into plasma cells are abundant, rough endoplasmic reticulum. The cytoplasm of the plasma cell becomes packed with RER because these cells are producing vast quantities, very large quantities of antigen-specific antibodies. If the animation doesn't run, simply launch the version of the same series of slides that don't have the audio files and watch the animation as well. To produce plasma cells, and in fact to elicit memory in B cells, these cells undergo what we call clonal selection and clonal expansion. What does that mean? A clone is simply a copy. Twins are copies or clones. And we have cells, B cells, that make copies of themselves. These are the B cells that recognize antigen. When we have a B cell that has just the right conformation of their antigen binding sites on their antibodies, and those particular specific or unique types of B cells, of which we have various different versions or all kinds of conformations or antigens that we can interact with, those activated B cells using interleukins in interaction with CD4 positive T cells start to become selected. These are the ones we want to trigger to undergo mitosis. That's clonal expansion. So another word for mitosis, or phrase I should say, that immunologists use instead of saying activating the B cells to undergo mitosis, we use the phrase clonal expansion. Because it's not just simply causing the B cells to undergo mitosis, but they're also being transformed, at least 80% of them, into plasma cells. So once again, our immune system and our specific immune response is very complicated. We produce about 80% activated plasma cells that are producing high quantities of antigen-specific antibodies, first in the form of IgM, and then we will undergo essentially a conversion from IgM to IgG to work and eliminate unwanted virus, toxin, or bacterial infections. 20% of those activated B cells do not transform into plasma cells. Instead, 20% of the activated T cells, B cells, transform into memory cells. I just about introduced the extra little bit of information just a second ago, in that not only do B cells undergo clonal selection and expansion, T cells do too. The difference being T cells don't produce plasma cells. But this idea of selecting out unique clones or unique sets of B and T lymphocytes that are antigen specific, and then expanding those populations for what we call very unique effector function. In other words, their job, what kind of work do they do? For clonal expansion of B cells, we produce plasma cells producing IgM and IgG with a pool of memory cells. And the same thing can occur with T cells, except that T cells become activated. They are going to expand or undergo selection for, again, the very specific subsets of Th1s or Th2s, and even further, more well-defined, smaller subsets have now been identified with very unique targeting capabilities. In each case, for both T and B lymphocytes, there's a small percentage, roughly 20%, of these activated cells that are sitting on the sideline in the form of memory cells, so that the next time we encounter the same pathogen, our bodies can mount a more rapid and powerful immune response called the anamnestic or secondary response. The secondary response occurs within maybe 24 to 48 hours. We have higher concentrations of immune factors, which can easily be monitored in the amount of antibody produced in an anamnestic response in comparison to the amount of antibody produced in a primary response. Notice that in a primary response, it might take up to 5 days for the primary response, or the action of T and B lymphocytes, to be essentially powerful enough to be detected, let alone provide any kind of protection. That's why within the first 3 to 5 days, or even 1 to 5 days of our initial infections, our primary defenders, our first defenders, are going to be the non-specific defenders. After about 3 to 5 days, then the specific factors, the immune system with its T and B lymphocytes with very powerful actions, then can undergo clonal selection and expansion and join the battle. Secondary anamnestic responses is why vaccines work, and vaccines are very effective